Welcome to Alter Health, the epigenetic code. This is where we talk about the science of how your life shapes your genes and how your body can change when you change the conditions around it. Real stories, real science, and real answers. Let's go. I want to try something different with you today. Most explanations of biology, the kind you find in textbooks and most podcasts, work from the top down. They start with a big system, then break it into smaller parts, and eventually you find out how the parts fit together. By the end, you have a structure in your head, but the structure is static. It does not move. It's a diagram. Today I want to invite you to follow something moving instead. I want you to follow a single molecule on its actual journey through the world and into your body, from the moment it begins as part of a plant growing in soil, to the moment it lands on a specific spot on a specific gene inside one of your cells and changes what that gene does. The journey is real. It's happening in some version right now inside your body with molecules from the food you ate yesterday or this morning. Most of biology, the part nobody told you about properly, is not about big systems. It's about millions of these small journeys happening in parallel every second of your life. Each one a little story. Each one a tiny decision being made about which of your genes will be louder today and which will be quieter. The journey we are going to follow today is the journey of one methyl group, a single tiny chemical marker that, when it arrives in the right place, has the power to silence a stretch of your DNA. When it doesn't arrive, the DNA stays loud. The marker is made from the food you eat. The journey it takes from your plate to your gene is one of the most important conversations happening in your body, and almost nobody outside of cellular biology has ever heard it described. I want you to come with me on this journey, and I will be your guide. We'll start in a field of spinach. Follow a single molecule into a bowl of salad. Watch it travel through digestion, through the bloodstream, through a specific enzyme that has a name many people will recognize from genetic testing, into a single cell, and finally to its destination on a specific gene. Along the way, you will see exactly where things go right, exactly where they go wrong, and exactly why so many people walking around today have what doctors call normal blood work, while their underlying methylation machinery is quietly running at half speed. By the time we are done, you will understand methylation in a way that almost no doctor will have explained to you. Not because you have memorized terms, because you have followed the path. This is Alter Health, the medicine nobody told you about. And I'm Dr. Nicola Schmitz, and today we follow a single molecule. Our molecule begins its life in a leaf, specifically a leaf of spinach growing in a field. It could just as easily be a leaf of kale or chard or romaine or beet greens, any dark leafy vegetable. But let's stay with spinach today, because the word for the molecule we are following comes from the Latin word for leaf, which is folium. The molecule is called folate. Folate is one of the B vitamins. The number it has been given in the alphabetical system of vitamins is B9. Plants make folate as part of their normal metabolism. They use it themselves for their own cellular work. And when an animal eats the plant, the folate moves into the animal's body, where it gets used for many of the same purposes the plant was using it for. Folate is, in a sense, ancient currency. The same molecule has been passing between organisms for hundreds of millions of years, doing the same kinds of work. Now, the folate in our spinach leaf is not exactly the form your body needs. The folate in plants is in a particular chemical configuration. It will need to be modified in several steps before it can do the specific work we are following today. But it carries, even in its plant form, the basic structure that the body will use. Think of it as a raw ingredient. The plant has provided the foundation. Your body will finish the cooking. Here's the first thing I want you to notice because it matters for everything that follows. The amount of folate in a leaf of spinach today is meaningfully less than the amount that it was in the same kind of spinach 40 or 50 years ago. The soils had been depleted by intensive farming. The breeding has favored size and shelf life over nutrient density. The same leaf carries, on average, somewhere between 20 and 40% less folate than its grandmother leaf carried when your grandmother was eating it. This is documented in the agriculture research, and it is one of the reasons that even people who eat their vegetables can struggle to get enough of certain nutrients. So our journey begins with a leaf that contains less of what we need than it used to. This is not a tragedy. The leaf is still nourishing, but it does mean that for a body to have plenty of folate available, it now needs to eat more leaves more often than it would have needed a few generations ago. So you pick the leaf, or someone picks it for you. It travels to a market, then to your kitchen, into a salad bowl, and then into your mouth. You chew, you swallow. The leaf, or what is left of it, descends into the dark internal landscape of your digestive tract. And here, our molecule begins the more interesting part of its journey. One more thing about folate worth knowing before we move on. Folate is light sensitive and heat sensitive. A spinach leaf that has been sitting under fluorescent lights at a supermarket for 3 days in a plastic bag has lost a significant portion of its folate before you even bring it home. Boiling water destroys folate further. By the time the leaf reaches your mouth, depending on how it was stored and prepared, it may have lost another 20 to 50% of its original folate content. Steaming or eating these raw and fresh preserves much more. This is one of those quiet details that nobody mentions when telling you to eat more vegetables, but it changes the math of what you're actually getting from them. Now let's go back. Inside your small intestine, the folate molecule meets the surface of your gut wall. To cross from the inside of your gut into your bloodstream, it has to be small enough and in the right form to pass through specialized transport proteins that sit in the gut wall like little doorways. Most of the folate from your spinach leaf will be transported successfully. But here's where the first complication can happen. If your gut is not healthy, if the wall is inflamed, if you have been taking medications that affect the lining, if there is dysbiosis in your microbiome, the transport can be less efficient. Some of the folate that should have made it into your bloodstream stays in the gut and is eventually excreted. The leaf gave you the molecule. Your gut decided how much of it actually got across. And this is one of the reasons that in clinical practice, 2 people eating the same diet can end up with very different folate levels. The diet is one input, the absorption is another. And many people with subtly compromised gut function are absorbing less of what they eat than the label on the spinach bag would suggest. Assuming our folate molecule makes the crossing, it now enters the bloodstream of the small intestine and begins its trip toward the liver. The liver is the next Mayer station, and it is where most of the heavy chemical work on the molecule happens. The folate enters the liver in a form that's not yet fully usable. It needs to be activated. And this is where we meet a piece of biology that has, in the last decade, become surprisingly well known to a small subset of people because it shows up on genetic tests, and the test results have sometimes been alarming. The enzyme that activates the full name is methylene tetrahydrofolate reductase, which is why nobody uses the full name. So MTHFR is the molecular machine that takes our folate molecule and converts it into the form your body can actually use to make methyl markers. Without MTHFR, the folate is stuck in an intermediate form. With MTHFR, the folate is activated and ready to work. Now, every human being has the gene for MTHFR. We all have the enzyme, but the gene comes in several variations, and some of these variations produce an enzyme that works less efficiently than the standard version. The most common variation reduces the activity of the enzyme by about 30%. A less common but more impactful variation can reduce it by closer to 70%. These variations are surprisingly common. Estimates suggest that somewhere between 30 and 40% of people in Western populations carry at least one copy of the less efficient version. A smaller percentage carry two copies, which produces a more pronounced reduction. This means that a meaningful fraction of the people you know walking around today are converting folate into its active form more slowly than the population averages assume. Our folate molecule in this story is fortunate. It passes through an MTHFR enzyme that is working well. The enzyme grabs the molecule, performs its chemistry, and produces the active form of folate called methylfolate. The methylfolate is now ready to carry methyl groups around your body. If our molecule had passed through a slower MTHFR, it would have taken longer and more of the folate would have been wasted along the way. This is one of the quieter reasons that many adults are functionally low in active folate, even with apparently normal folate intake. They are eating enough, but they are converting it more slowly. The blood test that measures total folate looks normal because total folate includes the inactive forms. The methylation machinery is running at 3/4 speed or half speed all the time without anyone knowing. Our newly activated methylfolate now has a job to do. It needs to deliver a methyl group, the small chemical marker we have been following, to the body's main methylation machinery, but it cannot do this alone. It needs partner. The partner is vitamin B12, specifically the active form of B12 called methylcobalamin. Methylfolate and methylcobalamin work as a pair. The methylfolate hands the methyl group to the B12. The B12 then carries it forward into the next stage of the journey. Without active B12, the handoff cannot happen. The methyl group sits with the folate, unable to move, and the whole system stalls. And this is one of the most important and least discussed partnerships in human metabolism. Folate without B12 is like a delivery driver without a vehicle. The package is ready, the transport is missing. Everything stops. And here's where another quite modern problem appears. B12 in food comes almost exclusively from animal sources. Meat, eggs, dairy, fish. The B12 in these foods is bound to proteins, and your stomach has to release it before your gut can absorb it. The release requires stomach acid. Many adults, particularly those over 50, have reduced stomach acid, either naturally with age or because they are taking acid-blocking medications for reflux or other conditions. Reduced acid means less B12 released, which means less B12 absorbed. And on top of this, vegetarians and vegans get essentially no B12 from food because plants do not make it. They need supplementation. And the supplementation to be useful for the methylation pathway needs to be in the active methylcobalamin form, not the cheaper cyanocobalamin form that appears in most basic multivitamins. So the system requires both folate and active B12 working together. If either one is in short supply or in the wrong form, the partnership breaks down. The methyl groups stop moving, the methylation pathway slows, and the slowing is invisible on standard blood tests, which measure totals rather than activities. Assuming our methylfolate finds an available active B12, the handoff happens. The methyl group is transferred. The B12 now carries it. The pair is ready for the next step. And the next step involves another molecule with a long name, often abbreviated as SAMe. The methyl group is loaded onto SAMe. You can think of as the universal currency for methylation in your body. It is the molecule that actually distributes methyl groups to all the places they need to go. To your DNA, to your proteins, your neurotransmitters, to the compounds that regulate your hormones. SAMe is the courier service. The methyl group is the package being delivered. Right now, the SAMe molecule with our methyl group attached is circulating through your body looking for a destination. Trillions of methyl groups are being distributed in this way every second. They are silencing genes, activating proteins, building neurotransmitters, regulating hormones, repairing damaged DNA. The whole system, properly running, is one of the largest information processing operations in your body, and almost none of it is visible from the outside. Our methyl group, riding on SAMe, now has to find the cell where it will be used. Let's say, for the sake of this story, that it's been directed to a cell in the lining of your blood vessels, an endothelial cell, the kind that forms the inner surface of every artery, vein, and capillary in your body. The SAMe molecule approaches the cell. The cell's membrane lets it in. Inside, our methyl group is unloaded from SAMe and handed over to one of the enzymes that does the actual work of methylation. There are many such enzymes, each specialized for a particular kind of methyl attaching job. Some put methyl groups on DNA, some put them on proteins, some put them on the molecules that regulate inflammation. Our methyl group in this story has been handed to an enzyme that puts methyl groups on DNA. The enzyme moves toward the nucleus of the cell. The nucleus is where the DNA lives, coiled and packaged, organized in specific architecture that allows different parts of the genome to be accessible at different times. The enzyme threads through the packaging, looking for a specific spot on a specific gene. The gene in question in this story is a gene that contributes to inflammation. When the gene is loud, it produces proteins that drive inflammatory signaling. When the gene is quiet, the signaling slows down. Your endothelial cells, like every cell in your body, have to constantly decide how much inflammation they should be generating. Too little, and infections cannot be fought. Too much, and the cells themselves get damaged. So the methyl group is attached. A specific cytosine, one of the chemical letters of DNA, at a specific spot in the gene now carries a methyl marker that wasn't there a moment ago. The marker is small. It's almost invisible against the size of the DNA molecule. It has consequences. With the marker in place, the gene is harder for other molecules to read. The proteins that would normally activate this gene have a harder time getting to it. The inflammatory signal from this particular gene gets quieter. Across thousands of similar cells, with thousands of similar markers being applied, the overall level of inflammatory activity in your blood vessels drops. This is what your body does billions of times a day in cells throughout your body. Methyl groups are applied, methyl groups are removed. The volume on different genes is turned up and down in response to what your body needs at this moment, in this tissue, in this cell. The whole system is dynamic. It's constantly being adjusted, and it depends every step of the way on the supply of raw materials we have been following. The methyl group has reached its destination. The story of this one molecule from a leaf of spinach to a specific cytosine on a specific gene inside an endothelial cell is now complete. But the work continues. The body is doing this trillions of times, and every single time the success of the journey depends on whether the materials are available, whether the enzymes are working, whether the partnerships are intact, and whether the conditions are right. I want to take a few minutes now to go through the journey again, but this time pointing out the places where it can fail, because in most modern adults, the journey is failing in at least one place, often in several. The first case it fails is at the field. If the leaf carries less folate than it should, the journey starts with less raw material. The body cannot make up for what was never there. The second place it fails is at the gut. If absorption is compromised by inflammation, medications, dysbiosis, aging, then less of the folate that was eaten makes it across. This is invisible from the outside, and it's rarely tested. The third place it fails is at the MTHFR enzyme. If the version of the gene you inherited produces a slower enzyme, the activation of folate runs at reduced speed. Across years, this adds up. Some people compensate by taking the already activated form of folate, called methylfolate, as a supplement which bypasses the slower enzyme. But most people never know they have a slower version because the standard tests do not check for it. The fourth place it fails is at the B12 partnership. If B12 is in short supply or in an inactive form, the methylfolate cannot hand off its methyl groups. This is increasingly common in older adults in vegetarians and vegans, and in anyone on long-term acid-blocking medication. The folate looks normal in the blood. The methylation pathway is still stuck. The fifth place it fails is at the SAMe pool. SAMe itself requires not just methyl groups, but also other nutrients to maintain its supply. If those are depleted, even a working folate B12 system cannot keep up. The sixth place it fails is at the enzymes that do the actual methylation. These enzymes can be slowed down by toxic exposures, by high alcohol intake, certain medications, and by chronic stress. The materials are there. The enzymes themselves are sluggish. And the seventh place it fails, which is in some ways the most important, is at the body's overall environment. The whole methylation system runs more slowly when the body is in a state of chronic inflammation, chronic stress, chronic sleep deprivation, or chronic nutrient depletion in other areas. Methylation is not isolated. It's one of the systems most sensitive to the overall condition of the body. This means that for most people walking around today, the methylation pathway is working, but it is not working at full capacity. The methyl groups are being delivered, but in fewer numbers, less precisely, with more delays. The fine-tuning of which genes are loud and which are quiet is happening, but less accurately. Over years, this drift contributes to almost every condition associated with aging, including cardiovascular disease, neurodegenerative disease, certain cancers, and the slow erosion of cognitive function. This is why, in the practice of working with the methylation system, we don't look for one thing. We look for the whole pathway. The folate intake, the B12 status, the MTHFR variants if relevant, the gut absorption, stress and sleep load, toxins, cofactors like magnesium and zinc and B6 that support the enzymes along the way. Fixing one piece without fixing the others rarely produces the changes people hope for. Fixing several pieces together often does. I want to take a few minutes to describe what the symptoms of impaired methylation actually look like in real people, because this is one of the most underdiagnosed patterns in modern medicine, and many listeners will recognize themselves in what I am about to describe. The classic picture is a woman in her late 30s or 40s, although it can show up at any age and in any gender. She's intelligent, accomplished, doing many things right. She eats reasonably, she exercises, tries to manage her stress. And yet, she has a constellation of symptoms that her doctor cannot quite name. Fatigue, but not the obvious kind. The kind where she wakes up tired even after 8 hours of sleep and where coffee no longer fully fixes it. Mental fog, particularly in the afternoons, where words come more slowly than they used to, where the names of acquaintances briefly disappear, where she walks into a room and forgets why. Mood swings that don't match what is happening in her life, with low patches that come and go without obvious cause. Sleep that is technically there but doesn't refresh, often with early morning waking around 3 or 4. Hormonal symptoms that her gynecologist calls perimenopausal, even when she's too young for that label, including heavier periods, breast tenderness, mood shifts tied to her cycle that didn't exist a few years ago. On the cardiovascular side, sometimes a marker called homocysteine, which the standard panels don't include, but which can be specifically requested, comes back elevated. Homocysteine is one of the intermediates in the methylation pathway, and when methylation runs poorly, homocysteine accumulates. Elevated homocysteine is one of the most reliable indicators of impaired methylation, and is also itself a risk factor for cardiovascular disease, for dementia, and for several other conditions. On the immune side, autoimmune conditions are more common in people with impaired methylation. The connection is biological. Methylation regulates immune gene expression. When the regulation drifts, the immune system makes more mistakes. On the cognition side, the slow erosion of memory and processing speed in midlife, which many people accept as normal aging, is often accelerated by impaired methylation. The brain is one of the most methylation-dependent tissues in the body. When the pathway slows, the brain is among the first to register it. Now, none of these symptoms are specific to methylation. They overlap with many other conditions. What makes them recognizable as a methylation pattern is the combination, the constellation. The woman who has 3 or 4 of these together, with normal standard blood work, with no clear cause and no clear treatment, is often a methylation case waiting to be identified. And the response to addressing the pathway, when it is actually the underlying problem, is often striking. Within weeks of supplementing the right forms of folate and B12, of supporting the cofactors, of addressing the stressors that deplete methyl donors, the symptoms begin to lift. The fatigue thins out, mood steadies, sleep deepens, clarity returns. None of it dramatic, but all of it real. And again, fixing one piece without fixing the others rarely produces the change people hope for. Let me give you briefly what the pathway needs to run well, not as a prescription, but as an inventory. Folate in the form of leafy greens, legumes, and some other vegetables. For people with slower MTHFR variants, methylfolate as a supplement which bypasses the enzyme bottleneck. Avoid the synthetic form, folic acid, when possible, because it's not the same molecule and it can sit at the enzyme without being properly converted, blocking the natural forms. Active B12 in the form of methylcobalamin, available in animal foods for omnivores and as supplementation for vegetarians and vegans. The cheaper cyanocobalamin form is less ideal. Vitamin B6, which is needed at several steps in the broader methylation pathway, available in many foods but commonly underconsumed. Choline, which is an underappreciated nutrient that supports methylation in a parallel pathway, found in egg yolks, in liver, in some legumes. Betaine, which is another methyl donor that backs up the system, found in beets, in quinoa, in spinach itself. Magnesium and zinc, which are required cofactors for many of the enzymes along the pathway, both are commonly depleted in modern diets. And the broader conditions that allow the system to run well. Adequate sleep, because sleep deprivation impairs methylation. Reasonable stress levels, because chronic stress depletes the materials and slows the enzymes. Low toxin exposure, because many toxins consume the very methyl groups the body would otherwise use elsewhere. Adequate protein, because amino acids are the building blocks of the system. And adequate calories, because methylation is energy intensive. None of these are exotic. Most of them are things we have discussed throughout this season in different contexts. The picture that emerges when you see them together is that methylation is the system that integrates almost everything else. Sleep affects your methylation, your food affects your methylation, your stress affects your methylation, and toxins do as well. The system is, in a sense, the meeting point of all the other systems we have discussed. And when methylation runs well, your DNA gets the regulation it needs. The volume on the right genes is turned up and down at the right times. Your cells get accurate information about what they should be doing. Your body as a whole organism runs with fewer mistakes. When methylation runs poorly, the regulation gets sloppy. The volume on certain genes drifts in the wrong direction. The body starts making decisions that are slightly off. And over years, the slightly off decisions accumulate into conditions that doctors eventually give names to. This is why the methylation pathway matters more than most people realize. It's not one thing among many. It is the thing through which almost everything else is translated into the body's actual behavior. There is one more dimension to this conversation that I want to bring in, because it changes how you think about methylation in particular ways. Methylation patterns are not just shaped by your own life. They are also, to a meaningful extent, inherited. The cells that became you carried, at the moment of your conception, a particular methylation landscape. That landscape was shaped by your parents' lives in the months and years leading up to your conception, and in some cases earlier. The Dutch hunger winter studies, which we discussed in the previous episode, were one of the first lines of evidence for this. Children conceived during that brief famine in 1944-45, who were exposed to the famine through their mothers' bodies during early pregnancy, showed altered methylation patterns at specific genes decades later. The methylation patterns were measurable in their adult bodies, and the diseases associated with those methylation patterns showed up in their adult lives at higher rates than in their unaffected siblings. More recent research has extended this picture. Maternal nutrition before and during pregnancy shapes the methylation patterns that the child will carry into adulthood. Paternal exposure to certain conditions in the months before conception, when sperm are being produced, also influences the methylation patterns transmitted to the next generation. The biology is not deterministic. The patterns can be modified by the child's own life, but the starting point is not blank. This means two things, both of which I think are important to hold. First, if you grew up in a family where the women had unexplained fatigue, where cardiovascular issues appeared in midlife, where cognitive issues appeared in later decades, where autoimmune conditions ran through the generations, the pattern you may be navigating is not just genetic. It may be methylation related. The pattern may have been shaped in part by what your grandparents experienced, and it may be modifiable in your own body in ways that the family history didn't suggest. Second, if you are planning to be a parent, the methylation environment you carry will shape what your children inherit. And this is not a guilt-inducing claim, it's a hopeful one. Working on your own methylation is not just for you. It changes what you pass forward. The children of parents whose methylation has been supported whose nutrient stores have been replenished, whose stress has been addressed, start their own lives from a different biological baseline than they would have otherwise. This is one of the more remarkable findings in epigenetic research, and one of the reasons I think the work of taking care of your own body is not just personal, it echoes. So that is the journey, from a leaf of spinach, through a gut wall, into the liver, through the MTHFR enzyme, into the partnership with B12, onto SAMI, into a cell, through an enzyme that places meta groups on DNA, to a specific spot on a specific gene that now runs a little more quietly than it did a moment before. This is happening in some version in your body right now, trillions of times, every second. The success of each journey depends on whether the materials are present, whether the partnerships are working, the enzymes are intact, the conditions are favorable. I want to give you a free resource this week because this episode covers ground that is hard to hold in your head all at once. In the show notes, you will find a guide listing the nutrients almost every Westerner is low in, why they matter for methylation specifically, and which foods carry the most useful forms. Print it out, keep it on your fridge, use it when you go shopping. The list is shorter than you might think. Next Tuesday, we go into the second largest input to methylation that almost nobody talks about: stress. Not stress as a feeling, stress as a biological force that depletes the very molecules we have been following today. We will look at how your nervous system, when chronically activated, quietly consumes the materials your methylation pathway needs to do its work. By the end of next week, you will understand why so many high-performing, successful, exhausted adults have what doctors call normal labs and bodies that are nonetheless quietly running on empty. If this episode opened something up for you, don't forget to subscribe and share it with somebody who has been told their fatigue is just aging. Many of those people have methylation pathways running at half speed, and they deserve to know that the pathway can be supported and that the support is not complicated. Thank you for listening, and see you next Tuesday, because health is a choice.